Okay, looks like people are slowly trickling in here. So I'm going to get started now. I'll keep admitting people as we go, but just to be mindful of time. Welcome everyone to the 2022 Euglena International Conference. Decoding the complete genomes of euglenoids, pathways to discovery, translation, and commercialization. My name is Scott Farrell. I will be your conference chair this year, and I am the VP of Innovation and Research Operations at Noblegen. So before beginning, I would like to thank our organizers, sponsors, and hosts, namely Kathleen Horlock-Roberts, Jonathan Holmes, Steve Barlow, Zubi Azima, and thank God Ebenezer. All of these people supported the conference and getting it off the ground, including all the promotional material you may have seen circulating, all the emails that we've been sending. Apologies for all spamming your inbox, but we do appreciate that. Also, our sponsors, including Jeff Horst from Kemen for supporting the photo contest and best paper awards for the EIN conference this year, and Noblegen for hosting the 2022 event. Thank you to all the attendees, the keynote speakers and invited speakers for filling out our excellent program of events. And just a little bit about the conference. So the conference will take place over the next 4 days and will cover the 4 major grand challenges, including basic biology, ecology and environment, evolution, and then finally translation and commercialization. On each day, there will be 2 sessions. After each session, there will be a panel period where we will have time to ask questions and have a conversation. This morning's session chair is Peter Myler. And without further ado, I'm going to hand it over to Peter to kick off the event. So thank you very much. Over to you, Peter.
Okay, thank you. Can everyone hear me and see me okay? I don't really care about the C part. Hearing would be important. Okay, so I'm Peter Myler. I'm from University of Washington and Seattle Children's Research Institute. I'm a bit of an interloper into the Euglena field. Worked on trypanosomes for many years. But recently got a little bit involved in some sequencing, primarily doing some high-C work with a graduate student of mine. So I guess what I should do is get started as quickly as possible. I'll just let the speakers go. I'll try to indicate when they have time. Maybe if you're a speaker, keep your eye on the chat, and I'll try to let you know when you have 5 minutes left. And then at the end of the time, we have a panel session. So I'll sort of try to organize that a little bit. And hopefully we can get finished in an hour and a half. The times here are in East Coast, US or Canadian time. And I'm on the West Coast, so I'll have to translate between one and the other. But anyway, without further ado, I'll introduce the first keynote speaker, who is Dr. Alison Smith from the Department of Plant Sciences at Cambridge University. And unfortunately, the PDF that I have doesn't have people's seminar titles, so I'd ask everyone to introduce their own title. So Alison, could you go ahead? So the next speaker is Natalia Gominska, and again, I apologize for mangling languages with which I'm not familiar. And she's at the Institute for Evolutionary Biology at the University of Warsaw in Poland. So Natalia, if you want to go over and take over the screen, and you have 15 minutes. I'm setting a timer. I will give you a warning after 15 minutes, and then if we can sort of wrap up after that without using up too much of the discussion time, that would be great. Thank you.
Okay, thank you very much. So I'm trying to share my screen with you. Are you able to see my presentation?
Yes.
Okay, so yeah, I am not from the Institute of Evolutionary Biology, rather from the Institute of Molecular and Cell Biology in Warsaw, and I'm going to tell you about the project I was involved in while I was doing my PhD research. This is my research grant, which was obtained from Polish National Science Center, and I'm going to present you the story about the repertoire of nuclear-derived circular extrachromosomal DNAs in Euglena gracilis. To wrap up, first I will introduce some terms which would be necessary to understand before we will dive more into the world of circular DNAs. Then I will present the diversity of this fraction of structural variants. I will tell some about how to investigate those types of molecules, what experimental protocol are involved in this type of research, what was the clue of the experimental protocol I was using in my research, and I will introduce briefly a bioinformatical approach I applied in this project, then present the results and wrap up the conclusions. So just without further ado, the ECC DNA are extrachromosomal DNA circles of endogenous origin. So basically, they are originated from nuclear or from other components of the organism's genome, and all of them, the whole repertoire of them, is referred to as circulome. It's similar to other terms like proteome in terms of, in case of proteins, or transcriptome in case of RNA transcripts. So the circulome is the whole, the entire population of eccDNAs from a given organism or a given cell. And the circseq is a type of analytical approach which can be applied to study those circulomes. Um, Maybe if thinking about the circular DNAs, first one can think about the plasmid DNA, which is typical to bacteria, or organellar genomes like chloroplast genomes, or maybe in wider context, the plastic genome, or mitochondrial genomes as well, or any other modifications of organellar genomes, which can be typical to certain taxa. Of course, this area, this repertoire is much wider. So the extrachromosomal DNAs may comprise of various classes of those circles. They can be circular variants of ribosomal DNA, and some organisms may have those as some sort of structural variants. Some have rDNA circles exclusive. Also, there are a lot of classes like SPC DNAs or microDNAs, which can be derived from repetitive regions as well, or from telomeres as well. Those are very diverse type of molecules. Some of them may contain some part of genes. Other of them can contain just repetition arrays. Some of them may be the carriers of oncogenes, and some of them are have some practical use. For instance, they can be used for screening for certain types of illnesses. And right now, nowadays, a lot of research groups have focused on those type of molecules, and they are under strong investigation in various types of organisms, beginning from the human to yeast and other model organisms, as well as more non-standard organisms, which prompted me to find out whether eccDNA repertoire is diverse in Euglena or not. Uh, to date, three types of eccDNA were postulated to be present in Euglena genome. Of course, all of us know about the chloroplast genome and rDNA circles present in Euglena. In some research, also mtDNA was prompted as a potential source of circular DNA. I put the asterisk by this class of circular DNA because some of the works indicate that mtDNA in Euglena gracilis is present only as a linear collection of particles, but in other research, authors postulated that they have spotted some circles. This is the interesting finding, so I wanted to figure out what is the real situation, what really happens inside Euglena. So basically, I used Euglena gracilis as my model organism for this project, and Euglena gracilis Z, which was obtained thanks to Professor Zakryś from my former lab, was my model for this this analysis. Well, as I said, the circular DNA can be investigated, the circulomes can be investigated in a whole genome scale. And to date, there are three main protocols which can be exploited, namely CircleSeq, CirculomeSeq, and CiderSeq. Those three pipelines basically have the same framework, which is the DNA isolation, then the whole DNA repertoire has to be lysed in a certain way to get rid of the linear molecules and to get rid of some other contaminations. So basically, there is an enzyme which is called plasmid safe ATP DNase, which can digest the linear molecules, but it omits the circular ones. So this enzyme was exploited in almost every protocol. Then, after the multiple rounds of digestion and purification, circular DNAs are amplified or segmented with special enzyme. The amplification is quite unusual because in those protocols, a special Templiphi polymerase is used, not a standard PCR cycles, but the rolling circle amplification, which leads to production of specific types of reads and enrichment of specific types of reads, which I will be talking about a little bit later. Then various sequencing platforms are used, either short read sequencing platforms like Illumina or long read sequencing platforms. Recently, I figured out that nanopore sequencing along PacBio could be used for this type of research. But when I was starting this project, only the PacBio was available. And since it was a little bit shortage of accession to PacBio in Poland, I was using the short read platform. After the sequencing step, of course, the bioinformatical analysis is required to collect, to interpret, and to filter the data. In my research, I used CircleSeq as a reference point to develop my own protocol. These protocols, which I was talking about, were developed while researchers were thinking about the model organisms, the standard organisms like yeast, like human or birds and other plants like Arabidopsis thaliana. But those are organisms which are highly studied, and there are a lot of pipelines, a lot of kits specially designed for purification and further manipulation of genetic material of those organisms. I had a more challenging task because Euglena is quite non-standard one. So I had to develop my own experimental protocol. Let's start from the experimental condition. I decided to use two different samples, differently treated samples. I had two cultures which were cultured in the same CM medium. And one of those cultures was kept in optimal photoperiod conditions, while the second one was subjected to UV irradiation. And ultraviolet is known as an agent which can cause DNA damage, and which is why the cell has to react involving some mechanisms of DNA repair. A lot of researchers previously postulated that ECC DNA formation may be the result of DNA damage or repair mechanism activity. So I wanted to check whether the conditions like UV irradiation or any other type of those conditions which can affect the molecular mechanisms of repair can prompt Euglena to produce more ECC DNA than optimal photoperiod. So, which is why I took those two conditions. Then I basically followed the principles of circle-seq protocol, but I decided to use two alkaline lysis steps before the exonuclease digestion and after that. This is why, this is because Euglena has a lot of secondary metabolites. Also, it has a very big genome, very unusual organization of genetic material. So I wanted to be sure that all of my linear molecules are completely or nearly completely digested. So I would not produce the artifacts or minimize the risk of producing artifacts, which could in turn affect my data analysis and lead to the false positive results. So after I applied this procedure, I obtained some sequences, I obtained some samples and then subjected them to sequencing and the rest of the analysis. And regarding the bioinformatic pipeline, you have to know that this is kind of tricky because in normal sequencing, the reads are mostly concordant, but those reads, which are usually considered as an artifact, as a trash, and are omitted from the analysis, are those droids we are looking for. So basically, there are some specifically designed approaches which can allow to filter the discordant or soft clipping reads and narrow the search criteria to find the reads which can pass through the potential circle breakpoints. So those were the reads which I was very interested in, and those were the reads I was looking for. Um, there are three main pipelines in bioinformatics which can be used to screen the genome and to find the potential circular DNAs. And two of them are suitable for Euglena. One of them was totally not suitable because the size of the genome of Euglena was too big. So first I produced the raw output with two different algorithmical approaches, then filtered the data according to quality criteria and merged them so I could have a comprehensive data set for two of those conditions. And here you can see the result of capillary electrophoresis. So yeah, in terms of the data integrity, the libraries were prepared according to the gold standard, and the quality of these libraries were also sufficient. So I will skip it up to make it quicker. And after the mapping, this is just one glimpse of the data. I don't want to force you to look through all of them, but basically most of the contigs were mapping to Euglena or to no-hit against the GeneBank database. But when I used Euglena genome, draft genome as a query, almost 99% mapped to the Euglena genome. So the library was prepared in a very good quality. And here are the results according to the size distribution of my ECC DNA candidates. What is strikingly easy to notice that both of pipelines produced slightly different sizes of those circular candidates. And this is quite normal because CircleMap is working mainly on a larger circular candidates, while ECC Finder allows to find smaller circles. So, which is why I combined those two to have the more comprehensive data set. And another thing which is quite unexpected, that regardless of the used condition, both samples were quite concordant. There was no actual big size difference between the Euglena which was kept in steady conditions or in Euglena kept under the UV irradiation. Most of the circular candidates were picked by a certain tool. I mean, most of them are tool specific. So this is another reason why I used two of them to produce more comprehensive results. And most of the short reads were found by a ECC finder, while Circle Map was better in finding larger ones. Of course, I found the rDNA. Especially interesting is that I've reconstructed two isoforms for a steady state, and for the Euglena, which was subjected to irradiation, three slightly different isoforms, and they are mostly, the differences sit in the repetitive regions. I also found some circular candidates which were derived from chloroplast, mitochondria, and from the nucleus. But as I can say regarding the nuclear genome, well, it's not that good in terms of the annotation quality. So I narrowed my search to the best known genes, which are deposited in a gene bank or other repositories. And then I screened them in terms of their origin. And what is strikingly interesting, that after applying some filtering criteria, just to show you here, I have filtered the candidates which were singular for a given reading frame. So basically here are only the most prevalent. And what was interesting, that I found some circular candidates which can confirm that mitochondrial derived circular RNAs, DNAs, sorry, are present in Euglena circulum. And most of them were present under the UV stress condition. To sum up my presentation, I've found out that circular DNAs have quite uniform distribution across genomic loci, and also they have a quite uniform distribution across the experimental condition. The differences between those conditions were very slightly, they were below statistical significance. So we can say they are uniform. Also, my screening allowed to detect the circular DNAs derived from the mitochondrial sequences. So right now we can confirm that mitochondrial DNA in euglenids may adapt the circular form. Also, those results are coherent with previous research, which was conducted on human genome or yeast genome, and recently on the pigeons. So basically, as in other eukaryotes, Euglena has a uniformly distributed components of its circulum. So basically, we cannot say that there are some regions specifically enriched in ECC DNA, and they've got some particular function in some bioprocesses, which can lead to a special formation of those type of molecules. Also, one has to keep in mind that this research needs to be treated with caution, because as Euglena doesn't have a well annotated, strongly curated genome, those findings could be treated only as some insights into Euglena circulum, not as the very exhaustive analysis. So basically, those are the main conclusions from my research up to date, and I hope they will be soon published, maybe in PLOS. And at the end of the presentation, I'd like to thank some people. First of all, I'd like to thank a few brilliant people from my former Institute of Evolutionary Biology, namely Paweł Hałakuc, who was my sparring partner and who provided me a moral support during my PhD, Professors Rafał Milanowski and Professor Bożena Zakryś, who were my PhD supervisors and who introduced me to fascinating world of Euglena. I also would like to thank people from my current lab, from Laboratory of RNA Biology, namely Paweł Krawczyk, who taught me to perform bioinformatical analysis, and Andrzej Dziębowski, who is my PI and who discussed the results with me very eagerly. Also, I'd like to thank Mariusz Czarnowski-Cieciura from Laboratory of Protein Structure, who gave me the opportunity to use the computational resources necessary for those analyses. And as I mentioned, this work was supported by the grant from Polish National Science Center. Thank you very much for your attention.
Thank you, Natalia. Scott has asked me to keep the questions for the end after the next talk. So let's go on to the next presentation, and then we'll come back for questions for Natalia and any of the other speakers. So the third speaker and final speaker in this session is Rodrigo Coloni, who's in the Laboratory of Molecular Enzymology at the Institute of Agrobiotechnology. My Italian's not good at, I think, of the seaside, and also in the Faculty of Biochemistry and Biological Sciences at the National University of the Littoral, which again, my Spanish is not good. So that's in Argentina. So Rodrigo, if you go ahead and perhaps you can tell us the English name of your institution.
Yes, thank you, Peter. I cannot share the screen because another one is sharing.
Natalia, maybe you need to stop sharing the screen, please.
Yeah.
You right? You good now?
Thanks, Peter. Yes, I am Rodrigo Caloni from the Instituto de Agrotecnología del Litoral from Argentina. Today, I'm going to talk to you about the characterization of two proteins from Euglena gracile. As you know, it's a protist organism with a wide metabolic capacity, able to grow in, able to grow in different culture conditions. Its reserve polymer is the paramylon, a beta-1,3 glucan, unbranched, that is crystalline and insoluble in water. In our group, we are interested in the study of enzymes involved in the paramylon synthesis and degradation. Previously, we have characterized the UDP glucose pyrophosphorylase, and also we study the role of this enzyme from the UDP sugar pyrophosphorylase in the content of the paramylon using RNA experiments. Regarding the degradation of the polymer, it is known that Euglena can consume it in conditions of energy need. And thanks to advance in transcriptomics and proteomics, we know that consortium of different enzymes would be required for the efficient degradation of this polymer. So recently, Robert Field's group reported the presence in Euglena of phosphorylase belonging to a new phosphorylase family. the glycosyl hydrolase 149. It has beta-1,3-glucan phosphorylase activity, and as you can see in the graphic, the reaction that catalyzes is reversible. So Robert Field kindly shared with us the plasmid, so we could expand the characterization of this protein. In the degradation sense, the phosphorylases reaction, we observe that as the polymerization increases, the catalytic efficiency decreases. And we did not observe activity against laminarin and paramylon. Using a 3D model, we design and characterize mutants in the active site. In this way, we find relevant amino acids for the enzymatic activity of the GH149 from Euglena. Also, we perform the confocal microscopy with specific antibodies for this protein. And we found recognition signal in both culture conditions, autotrophic and heterotrophic. And as you can see, we can see hotspots between the organelles and the paramylon granules in both types of culture conditions. We are also interested in the study of enzymes with endoglucanase activity in Euglena gracili. So we decided to study the glucanase of the family 64, of which there was no further information that is mRNA sequence. So the gene coding for the enzyme was heterology expressed. We made a two-step purification, after which we got the GH64 in a soluble form. We made a 3D model where we observed the two classical domains of this family, the red and the blue. The blue is the catalytic one, and the pink domain with beta sheet has not been observed in another GH64 from another organism. This enzyme has the characteristic crescent-like form of the endoglucanase with a zone of high electronegativity important for the binding of the substrate. The active site has aromatic residues to stabilize the incoming polysaccharide chain, and has an additional aspartic acid that has not been observed in another GH64. So we perform a functional characterization of these enzymes. The optimal reaction conditions that we observed were 4.5 pH and 15 degrees. We characterize the thermostability, and we see that it remains 80% of the activity until 50 degrees. We also determine the effects of metal ions on the activity of the enzyme. Well, we also perform TLC to characterize the hydrolysis products of this enzyme. The small product that we see was the pentasaccharide, the laminary pentose, as the small product. So we decide to characterize this protein kinetically against these two substrates. Also, against the beta-glucan from sheep. So we observe a better catalytic constant for the laminarin and a higher affinity towards the cis beta-glucan. No? So when we analyze the catalytic efficiency, as you can see also in the graphic, the enzyme was at better efficiency against the cis beta-glucan. So because we use two insoluble substrates in this analysis, we decide to complement this with a kinetic model prepared for insoluble substrates. So we calculate the gamma parameter that briefly tells about the density of enzyme attack sites on the polymer. No, how many sites could find this enzyme in the polymer. So what we could see is that the enzyme finds more sites to attack in the cis beta-glucan than for the other two substrates. So with these results, we were interested in analyze what is the effect of incubate this enzyme with a shift. In our case, we work with Cluberomyces lactis, and we observe that as we increase the CH64 concentration, the number of colonies of Cluberomyces lactis increases, as you can see in the graphic. Well, this is our team, our institute. I would like to thank our funding support, the CONICET, Agente Nacional de Promoción Científica y Tecnológica, la Universidad Nacional del Litoral. And in the name of our group, I would like to thank to all of you, and especially to the organizers of this international meeting. So, thanks.
Thank you very much, Rodrigo. Thank you for keeping us on time. So I think at this stage we should open the session up for panel discussion. So any questions for any of the speakers? I do see a couple in the chat already for Natalia, so maybe we could start with those. The speakers who put those in the chat could either read them out themselves, or maybe Natalia, you could read them.
Unfortunately, I don't have a possibility because I cannot see them.
Oh, okay. All right. I'll read them then. So the first one was from Sridharan Govindachari, and that was whether the question was whether there was any benefits of making circular molecules under stress conditions.
Well, according to modern findings, those circular DNAs are rather the byproduct of DNA damage than they have a certain particular functions. They may act rather against the cell, for instance, as oncogene carriers in some instances, not particularly in Euglena, of course, because it's understudied, but in human, for instance, or in birds like pigeons, those can act as agents which can be responsible for illnesses. Also, on the mice model, there was a study regarding Alzheimer's, and the researchers, the authors of this study, found out that some certain circular isoforms may be responsible for increased risk of having Alzheimer's.
Okay, thanks. I might add something there from my experience in Leishmania, where there are, of course, circular molecules. And the evidence in Leishmania, mostly from the French group a few years ago, indicated that Leishmania does, in fact, have an advantage of making circular molecules, in that they constantly make these circular molecules that will be expressed sort of randomly, but then under certain conditions, some of them will be selected. So under drug conditions, they select circles that are amplified and give them a selective advantage. So it's interesting that since Euglena is sort of related to the trypanosomatid, whether there might turn out to be some advantage to Euglena, but obviously we need to do a lot more work on that. So second question was from Vladimir Humpel, who wonders whether the nuclear genes that are present in the circulome contain introns, and I guess I would add whether they're the same introns as you would find in the circle, or is there a bias towards some type of introns?
Well, regarding the intronic sequences, of course, there are a lot of circles which can be produced by intronic sequences, especially that in euglenids there is Euglena typical type of intron, which are called non-conventional ones, as you may know. And those introns have a non-canonical borders, which are very rich in repetitions. So basically, every part of the genome which is rich in repetitions may serve as a hotspot for ECC DNA production. So those introns are more eagerly incorporated into ECC DNA, although there are some constraints in studying them because some of intronic sequences, repetitive sequences, are found to be present in many various introns, which are widespread throughout the Euglena genome. So basically, it's hard to say whether they are mapping into the certain genomic loci or whether they are multi-mapping. Mostly they are multi-mapping, and most of those circles are derived from non-conventional introns. Although I've spotted some circles with mixed composition, namely containing some part of exonic sequences, some part of intronic sequences.
Okay, thank you. Scott, I see you have posted a question. Perhaps you could ask that directly.
Yep. My question for Alison was if she had tried expressing these pathways or specific enzymes of these pathways in other organelles to take advantage of some of the other effects, but she's answered the question, and she said that when you do try expressing these in other parts, for example, the cytosol, you get very little production of the products. So thank you.
Yeah.
Thank you for.
I think just to say, we were targeting the enzymes of the chloroplast because that's where GGPP, which is the precursor to make the diterpenoids, is made in abundance. So it would be difficult to, you'd have to try and engineer production of the precursor in another organelle. And actually, we think I don't think some compounds might be toxic, but actually the main issue is there's a growth penalty there appears to be a growth penalty for producing these compounds, just sort of overall. So it's not that the cells are dying, it's just presumably a metabolic load that comes from funneling GGPP through to the diterpenoid rather than to the primary metabolites.
Got it. Thanks.
Okay, I see one more question from Hala. I can't see the last name. And that was whether circle-seq has been used in other microalgae. So this was obviously a question to Natalia. So
thanks for this question. Well, to be honest, I was the first madman who tried to apply this technique to microalgae, and as far as I know, there were no other research groups which wanted to do so.
okay, thank you, Natalia. So any more questions? I see. Oh, Vladimir, you have your hand raised. Did you get your question answered, or do you have another question?
Hello. Yeah, I actually have addition to my question. Can you hear me?
Yes, we can. Go ahead.
Hello, Natalia. Actually, in my question, I was more interested whether the nuclear encoded genes that you found in the circulome, because I understood that you see some genes encoded in the circles, right? So whether these carry the same introns as you see in the genome, or whether the introns in these tubulins or other genes that you have found there are anyhow, you know, they lose the introns while they are going into the circles.
Okay, to answer this question, I have to clarify a little bit that those circles are not encompassing the entire open reading frames. They have some sort of a proportion of these reading frames. Usually they are fragmented, so they can have a few complete exonic regions, then followed by a normal intron. Sometimes there are mismatches, but I am unable to say without the experimental verification whether those are sequencing artifacts or are those really extended or shortened isoforms. Basically, if they are the byproducts of genomic rearrangements, they may be some sort of defective ones, which were excluded from the genome in the first place. So basically, there is a little bit of diversity among them, but none of them is a complete open reading frame.
Oh, okay. Thank you. Thank you for clarification.
You're welcome.
Okay, I might ask a question of Alison, and that is, what is the value to you? And I think I know the answer, but maybe you could give us a better answer of having a good annotated genome sequence, and is that really an impediment to using Euglena at the moment?
Well, I think, as I said before, metabolic engineering per se is very empirical. You know, you have to try different things. It's not obvious how you can manipulate. We can't design processes, unfortunately, as you can in other hosts like yeast or E. coli. And my experience of those is actually that they can also be quite challenging. So the more information you have about your organism, the better. I do know that the gene structure in Euglena is incredibly, is unusual, shall we say? It's not canonical, and therefore having an understanding of the endogenous promoters and the trans-splicing of the 5' UTRs and intron structures and so on, all the things that you have to have will certainly help in trying to express genes, because actually getting the gene in is only the first part of the metabolic engineering. So the more you know, the better. And I don't know whether you need, if you're trying to manipulate endogenous metabolism, increase the production of secondary products or knock something out and so on, that might be necessary to know about the metabolism or to know about the genes, specific genes. But certainly knowing something about how genes function, whether introns are important, whether you need to have flanking sequences or chromatin associated regions and so on. I'm sure it would all be helpful. However, most metabolic engineering, as I said, even in yeast, I think, even in bacteria, is a bit like, oh, I've got an enzyme or I've got some enzymes, I'll put them in and see what happens, and then I'll tweak it.
Okay, thank you very much. Does anyone else have any more questions? If so, raise your hand, or if I can't see you, just open. Michael, you have
Peter,
something?
I was just going to suggest, could we cut to the break now so that we can begin at 1:30? And any other questions we can push back to our next panel session, which we might have a little more time as a buffer to answer.
Okay. Okay, that is fine with me. So thanks, everyone. Thanks to the three speakers. Very interesting talks this morning. So yes, I think we should break and have a short break where we're a tiny bit behind our original schedule. So I think if we can have a 5 or 10 minute break and get started at 8:30 in my time, which whatever time you are in the world, you'll have to figure that out, or slightly after that. So again, thank you very much, and I look forward to the remaining sessions.
Thank you, everyone. We'll see you in 5-10 minutes. Thank you, Peter.
Professor Saul, are you here and ready? Just checking before we begin.
Yes, I'm here and I'm ready.
Wonderful. We'll start in a minute.
Okay.
All right, welcome back everyone to our second session for day 1 of basic biology of Euglena. So I will now throw the session over to Professor Saul Purton from the Department of Structural and Molecular Biology, the University College London, who will be talking about genetic engineering of the Chlamydomonas chloroplast as a model for euglenoid plastic biotechnology. And I'll just say, similar to Paul before, I'll give you a message at 5 minutes when you've got 5 minutes remaining. And for those of you, write your questions in the chat and we can go over them at our final panel session. Saul, it is all yours.
Okay, so thank you, Michael. Can you see those slides?
Yes, we can see them.
Excellent. Let me just pull up a pointer. Okay, so my talk is sort of continuing nicely from Alison's in that I'm also going to talk about the genetic engineering of Chlamydomonas, but I'm going to focus on the chloroplast. And hopefully, if I have time at the end, I'm going to talk about how what the opportunities and challenges are of transferring the technology we've developed for engineering the chloroplast of Chlamydomonas into Euglena. So as Alison explained in her talk, algae have the potential to be exploited as low-cost and light-driven platforms for making recombinant products. And those recombinant products can be high-value proteins, things like pharmaceutical proteins, such as antibodies and vaccines, etc. They could be biopolymers for various different applications, or as Alison talked about, they could be metabolites such as terpenoids or oils or vitamins that have value. And given that we now live in the age of synthetic biology, we have the potential to use in these molecular tools to rapidly engineer our platform to make this, a whole range of different products. And obviously what we need in terms of an algal platform is one that is genetically tractable, where the basic technologies for transformation have been developed, one that's easy to grow, particularly at scale, and ideally one that's edible or at least non-toxic. And as Alison explained, really at the moment, it's algae such as Chlamydomonas and Phaeodactylum that are being used to explore this potential. But potentially in the future, certainly if the molecular genetics can be improved for Euglena, then Euglena obviously offers an attractive industrial platform for making high-value recombinant products. So as I say, our focus is on engineering the chloroplast and using Chlamydomonas to do that. Chlamydomonas is particularly attractive because it contains just a single chloroplast that fills about half of the cell. And within this subcellular compartment, you have a very simple genetic system. And in Chlamydomonas, you have a polyploid circular genome that's about 206 kilobases. And as it says down the right-hand side here, it uses the standard genetic code. So unlike other many other organelle genomes that deviate to some degree from the genetic code. PAMI uses a standard genetic code. Within the genome, it encodes all of the RNAs that it needs, so a set of 26 tRNA genes and 4 ribosomal RNA genes. So in that sense, it's a self-contained system. There's no RNA import into the chlamyd chloroplast. Of the protein coding genes, there are 69, and because chlamydomonas can dispense with photosynthesis and grow heterotrophically on a carbon source, then all of the photosynthetic genes, which is 43 in total, are essentially non-essential under those heterotrophic conditions. It's also a nice sort of simple plastome or chloroplast genome compared to other organelle genomes. There's no overlapping genes, there's no RNA editing, there's only 7 introns scattered throughout the genome, so very few introns. And transcription is simple in the sense that it just uses a single type of RNA polymerase, unlike higher plant chloroplasts, and a single sigma factor, unlike many other algae. Translation is on a eubacterial type 70S ribosome. And as an extra bonus, chlamydomonas has a simple sexual cycle. So any engineering that, for example, Alison does in the nuclear genome and we do in the chloroplast genome, we can actually combine those genotypes just simply by carrying out a simple cross. So it represents, this compartment represents a nice subcellular platform that potentially we could program to make a whole range of different things. So over the years, my group and others have been sort of improving and developing the tools for chloroplast engineering in chlamydomonas. And we now have quite a nice short transformation pipeline or workflow where we can build a construct, and I'll talk about this a little bit in a minute. We can build a construct using Golden Gate methods that contains the transgene we want to introduce. We can introduce it into the chloroplast using one of 2 methods. We can either use a gene gun and essentially bombard a lawn of algae with gold particles that have been coated with the DNA, or we can actually use a sort of very simple, cheap, poor man's alternative, which is to essentially take a suspension of cells and plasmid and glass beads and just vortex the whole lot together. And the abrasive action of the glass beads creates transient holes in the cell membrane and the chloroplast membrane, allowing the DNA in. And that latter method is illustrated here in that we've transformed an antibiotic resistance gene into the chloroplast. You can see we actually don't get that many colonies, but as I'll show you, that actually doesn't matter because in principle, all of these should be genetically identical. So we only need a handful of colonies just to check that the transformations worked. From those colonies, we can pick them off and obviously restrict them on solid media to produce enough for molecular analysis. And we've developed a very simple 3-primer PCR test to actually make sure that not only has the transgene gone in, but it's gone in the right place. And also that the chloroplast is what's called homoplasmic. That is, all of the 80 or 90 copies of the chloroplast genome have now acquired the transgene. And basically, if we see this upper band here and we see the absence of this lower band here, then that essentially ticks all of those boxes. We can then check at the protein level that the protein's being made. Very often we put an epitope tag like a hemagglutinin tag or a streptabalin tag onto the protein so that we can use commercial antibodies to check for production of the protein. And then we can scale, and this is our sort of simple little production facility within our lab. These are 20-litre hanging bags, and you can see that we can go from plates to 80 litres of algae, and then harvest that and dry that for analysis. So like Alison, and actually in collaboration with Alison, we've developed a a second Golden Gate assembly method specifically for the chloroplast, where, as Alison described, we can take individual parts, promoter, ribosome binding site, the coding sequence of gene of interest, etc. And in a single pot, using a single restriction enzyme and ligase, we can assemble a plasmid that has all of the parts assembled in the correct order and in the correct orientation to give us a functional gene, as shown here, together with these flanking regions. And these flanking regions are derived from a target region from within the chloroplast genome, so that homologous recombination between the flanking regions and the equivalent region on the chloroplast genome results in the targeted insertion of our gene and the creation of a stable integrant. So unlike nuclear transformation in Chlamydomonas, as Alison was saying, where the DNA goes in randomly, here we have precise insertion of our transgene into any place within the chloroplast genome. So we can essentially do what's called DNA surgery, where we can specifically open up the genome at one point and drop in our gene. So the Golden Gate assembly method we use, we've called it STEP, or Synthetic Toolkit for Engineering Plastomes. And again, as Alison described, it's based on using these type 2S enzymes that essentially allow us to have a library of individual DNA parts that we can combine together to create a transcriptional unit. And with our system, we use SAP1 as the key enzyme, and this gives a 3 base overhang. And the reason that that's important is that it means that we can create a perfect fusion without any additional DNA at the ATG and at the stop codon. And particularly around the ATG, that's quite critical in terms of allowing efficient initiation of translation. So we don't end up with any DNA scars when we assemble our transcriptional unit. And then we can go to a higher level, level 2, where we take multiple transcriptional units and string these together with our flanking sequences to build our final construct, our final plasmid for transformation. And this was all, this particular Golden Gate technology was based on a similar method that was developed by John Heap for bacteria. And he was very helpful in terms of helping us develop that. We've also made the whole process as simple as possible. At this level 1 stage, there are actually 11 different possible vectors that you can build this transcriptional unit into. And so to avoid mixing them up, we've actually, if you like, labeled each of those vectors with a different chromogenic protein. And each of these chromogenic proteins, or the genes for these, are replaced with our transcriptional unit during the cloning. So for example, instead of getting a red colony for just the vector, you now get a white colony. So we have a very nice way of scoring for the correct insertion of our constructs into any of these vectors. The other thing that we wanted to do, and this was with an eye to, well, both addressing potential regulatory issues to do with making transgenic algae, and also wanting to be able to do multiple transformations of the chloroplast, is to avoid ending up with transformants that have an antibiotic resistance marker in the genome. So we developed this method for creating marker-free transformants, and this is based on a 2-step system, or a positive-negative system, whereby we can select, using this spectinomycin resistance gene, AADA, we can select initially for the uptake of the transgene construct, but then we can actually loop out this whole region, because we have 2 features here. We have a direct repeat of this terminator region, this RBCL region, and we have a gene here which confers the negative selectable phenotype. That is, if this gene is being expressed, then the cells are sensitive to a compound called 5-fluorocytosine, or 5FC. So when we plate these cells on 5FC, it encourages the recombination, the intramolecular recombination between these RBCL repeats, and the looping out of this whole region. So we end up with transformants where the only thing we've inserted is our gene of interest. And obviously we can go around this cycle again and put more genes of interest in, reusing this POSNEG marker. So that whole process takes about 2 weeks to generate these marker-free transformants. So just some examples of testing the system. One of the early things we tried producing in Chlamydomonas was human growth hormone. It's a nice, simple, single subunit protein with no cofactors, and the only post-translational modification is the formation of 2 disulfide bonds. So you can see here from this Western analysis that we are producing human growth hormone. It's about 0.5% of the total soluble protein that is our recombinant protein. But even at that low level, we can demonstrate that the protein is folding and it is active. So this is a growth assay using a mammalian cell line that will only grow and divide in the presence of human growth hormone. And you can see that just adding a crude algal extract, you get a promotion of growth of this cell line, whereas the control algal extract has no effect. So an early demonstration that we can make a pharmaceutical protein and a functional one and actually be able to demonstrate it even in just a crude cell lysate. Other things that we've been making in Chlamydomonas include antibacterial enzymes that target particular bacterial pathogens. So these two here, CPL1 and PAL, are 2 that target Streptococcus pneumoniae. And these are lytic enzymes derived from bacteriophage that specifically target Streptococcus. And again, we can show that the algal extracts are highly active. So this is what's called a turbidity reduction assay. Basically, we're taking a thick broth of Streptococcus pneumoniae and just adding the crude extract to it and monitoring the cell's lysine as a drop in OD, in optical density. So this is a control. This is a chemical lysis of the cells. And you can see that, for example, for the PAL extract, within about 60 to 80 minutes, again, we've effectively killed all of the bacteria within the broth. And we can confirm that by actually plating samples out after 80 minutes. And both for PAL and for CPL1, there's no colonies that appear. So these lytic enzymes are incredibly effective, even as a crude extract, in terms of completely killing a thick, all the cells in a thick broth. And then just one third example, the idea of using algae as oral vaccines. So if we can reduce an antigen in the chloroplast of the alga, and then from the harvested cells, formulate those into the animal feed, then we can actually have a very low cost way of delivering vaccines to livestock, to fish, to shellfish, to poultry, etc. And this is just one example. This is a Chlamydomonas strain where we produced the major envelope protein called VP28 from a viral pathogen called white spot syndrome virus that has a devastating effect on shrimp or prawn production. And in this case, even though we got very low levels of production of the vaccine, just formulating this feed, this algae into the shrimp feed had a profound effect. So they were fed prior to the viral challenge, either with controls or with the cells containing the vaccine. And you can see that the cells containing the vaccine confer quite a significant protective effect. So up the side, this is mortality. So in the absence of the vaccine, you're getting almost 100% mortality within 5 days or 8 days of being exposed to the virus. Whereas the shrimps fed on the feed containing the vaccine algae are protected down to a level of sort of 17% or something mortality. So we think there's a real potential for using algae as a low-cost platform for making these sort of oral vaccines. looking at my time. Okay, so a couple of other things that we've done in terms of developing the technology is to think about both transgene biocontainment and crop protection. So obviously, if you're imagining growing genetically engineered algae at scale, then you have concerns, first of all, in terms of their containment. It's relatively easy to build in containment of the algae themselves, but how do you stop the transgenes from moving to other organisms through horizontal gene transfer? And also a second issue is, how do you stop this thing becoming contaminated? So using these low-cost photobioreactor systems, they are prone to contamination. So we've addressed both of these, or at least tried to address both of these. The first one actually came out of a completely unrelated problem that we had, which was that for some of those antibacterial proteins, we were having problems cloning the genes in E. coli because there is low-level expression of our constructs in E. coli. So the chloroplast promoters tend to work in E. coli. And so we ended up killing the E. coli cells and not getting any clones. The way we solved it was to take advantage of the fact that in Chlamydomonas, in the chloroplast genome, none of the protein coding genes actually use this stop codon, UGA. So there's 69, 65 use UAA, and 4 use UAG. And so this codon could be reassigned to encode anything without affecting any of the endogenous genes, because none of the endogenous genes is dependent on this being a stop codon. So the strategy was this, that we would take our problematic gene and identify, for example, a couple of tryptophan codons and change those tryptophan codons to the stop codon. And that way we would not have this problem of killing the E. coli, because this gene wouldn't be translated fully in E. coli. And if we also introduced, when we did the transformation into the chloroplast, an additional gene for a tRNA encoding tryptophan, but where the anticodon is now been changed so it recognizes TGA as its cognate codon, then in principle, we should be able to make the protein. So basically the modified tRNA here should read through these two stop codons and give us a full-length protein. And actually that worked really nicely. First of all, the system allowed us to clone these problematic genes in E. coli, because not only did the gene with the stop codon itself not get translated, but the addition of the tRNA gene and its expression in E. coli doesn't actually rescue the or read through these stop codons. So there's no expression of the transgene in E. coli. And also another thing that we were concerned about is whether this tRNA would actually interfere with the native system in E. coli. And again, it doesn't, so there's no effect. However, in Chlamydomonas, we get really nice translation of our gene when we have both parts of the system. So the gene itself with the two stop codons and this additional modified tRNA. And again, even though we have this modified form of the tryptophan tRNA together with the native tryptophan tRNA in the Chlamydomonas chloroplast, again, there seems to be no effect on growth. Again, we were concerned that this would maybe interfere with reading of bona fide tryptophan codons. So that solved our problem of cloning toxic genes, and indeed we sort of proved it by taking some highly toxic genes that actually have proved to be very problematic in terms of cloning in E. coli, and we were able to build constructs because we put these stop codons in and get them expressed in the Chlamydomonas chloroplast. So this gave us a system to allow the cloning of toxic genes, but at the same time, it also provides a biocontainment of these transgenes. So if this gene or if this whole gene cassette was to escape into a bacterium, for example, then although it might be expressed, it wouldn't be fully translated and therefore not give rise to a functional gene. So we have a nice way of at least providing a reasonable level of biocontainment of transgenes. And then the second issue was this idea of crop protection. And by that, I mean preventing your crop of algae becoming horribly contaminated with all the weeds, all the bacteria and the fungi that would otherwise grow in this rich media. And there's a practical aspect of this, which is that there's a cost associated with preparing large volumes of sterile media, and there's the challenge of actually just setting up and running these things without contaminating the cultures. So what we did was to use or exploit an aspect of sort of fundamental biology, which is that eukaryotes and most prokaryotes are reliant on phosphate as their source of phosphorus, and they can't use phosphite as an alternative. However, there are enzymes in a few bacteria that are able to convert phosphite, so PO3, into phosphate. And one of these enzymes is called PTXD. So we reasoned that if we put this enzyme into the chloroplast, then Chlamydomonas would be able to grow in a media containing phosphite, because it would be able to convert it into phosphate. Whereas all the other contaminants wouldn't be able to utilize the phosphite. So we expressed the PTXD in Chlamydomonas and analyzed the growth performance of the PTXD transformant compared to the wild type. And basically in phosphite media, you can see that the wild type doesn't grow either under mixotrophic conditions or phototrophic conditions, whereas the transformant grows quite happily. Indeed, it grows almost as well as if it's in regular phosphate medium. So essentially, the Chlamydomonas is just converting all of the phosphite it takes up into phosphate. So we now use this to basically go back and re-engineer some of our transgenic lines. So we've put the PTXD gene into, for example, a transgenic line that's making a vaccine against a fish virus called nervous necrosis virus. So we have a double transformant here that makes the antigen or the vaccine and PTXD. And just to demonstrate how well this works, so this is a regular hanging bag with phosphate medium, and we haven't autoclaved or sterilized the phosphate medium. In fact, we deliberately contaminated both these bags with a cocktail of fungi and bacteria. And you can see with regular phosphate medium, you get this horrible contamination. The bag is absolutely full of fungi and all sorts of bacteria. Whereas in the phosphite medium, there's almost no detectable contaminants. So even in non-sterile medium where we've deliberately tried to contaminate it, only the algae grows. So we think this is potentially a very useful tool for large-scale production of the algae. 2 more minutes, Michael. Yeah.
We've got a small buffer of time, but yes, if you could slowly wrap up, that would be appreciated.
Right, okay. So just to finish off, I just wanted to sort of talk about how about the possibility of transferring these technologies to other algae, such as Euglenoids. So here's the chloroplast genome of Euglenoids, and this was sequenced many years ago, back in 1993 by Rick Halleck's group. It's slightly different from the Chlamydomonas genome. The gene complement is similar, but you have this unusual structure where the genes are, there's a polar arrangement to the transcription, where most of the genes on the top here are arranged into operons that are transcribed anti-clockwise, and then on the bottom of the genome, they're all transcribed, or most of them are transcribed as operons in the clockwise direction. And also this genome is absolutely full of introns. So there's about 150 introns here compared to 7 in the Chlamy genome. So say similar gene content, but the arrangement of these genes is quite different. So what are the opportunities and challenges for developing chloroplast transformation for Euglena? Well, the strengths of Euglena are that, first of all, it's a large cell, relatively speaking, compared to other algae and Chlamydomonas, and also it lacks a tough cell wall. I think essentially it's a naked protoplast with a sort of thin protein pellicle. So in terms of delivering DNA into the chloroplast, this is quite an attractive target. Like Chlamydomonas, it's capable of heterotrophic growth, and chloroplast mutants have been reported. So there is potential there to use those chloroplast mutants as recipients and use the chloroplast genes as selectable markers to restore photosynthesis. There was, again, from the Hallett group, an early report of chloroplast transformation using the spectinomycin-resistant gene that I mentioned. But there are a number of issues as well that need to be addressed. First of all, you've got multiple chloroplasts per cell, I think something like 10 in a typical Euglena cell. And you've got 3 membranes rather than 2 around the chloroplast, which may present an additional barrier. That early report of chloroplast transformation couldn't show that the transforming DNA was integrating into this genome. And one of the sort of concerns in terms of developing chloroplast transformation for non-green algae or non-chlorophyte algae is that homologous recombination doesn't work or doesn't work effectively in these systems. And as I say, you've got this rather unusual gene structure. Not only do you have these multiple introns and these extensive operons, but there's an extreme AT bias. I think the genome is something like 74% AT compared to about sort of 64 for Chlamydomonas. And that complicates transgene design and codon optimization, and also knowing where to land your gene into this genome. And then finally, as far as I'm aware, there's no sexual cycle for Euglena. So again, that complicates or makes it more difficult in terms of wanting to combine different transgenic lines into a single line. So yeah, I'm going to sort of leave it there. If anybody is interested in talking further about developing chloroplast transformation for Euglena, I'd be happy to talk to them offline. So I'll just stop by acknowledging all of the people that basically done the work that I presented, sort of past and present members of my group. Alison Smith, so we have a long-standing and ongoing collaboration to develop further chloroplast genetic engineering in Chlamydomonas. John Heap, who helped us with the initial development of the STEP technology, and Tanya Wanathong, who has helped us with the vaccine work. So I'll stop there and happy to take questions.
Great, thank you, Saul, for that very informative presentation. I'll ask you just to stop sharing your screen now. So if you have questions, please just log them in the chat and we can read them at the end of the panel. I'll now welcome Dr. Sahachai Ingwangwon of the Chiang Mai University, who will be talking about quirky carbon metabolism in Euglena gracilis.
Yes. Okay, well, everyone can see my presentation?
Yes, we can see your screen.
Okay, cool. Hi, everyone. My name is Sahachai Ingwangwon. Now I'm lecturer in System Biology from Chiang Mai University, Thailand. Today I'm going to talk about the carbon metabolism in Euglena gracilis, which is my PhD thesis that I did at Department of Plant Sciences, University of Oxford, with Professor George Radcliffe, Professor Nick Kruger, and Dr. Alice O'Neill. The theme of today's meeting is the decoding of complete genome of Euglena, emphasize that the complexity of its genome, right? And this complex arose from the very complex evolution of Euglena as well, that expect to be multiple of endosymbiotic events, both primary and secondary. And without the complete genome of Euglena, it's very difficult to understand the metabolic capacity or the metabolic potential of it. There's other technique that can be used as well, including the transcriptomic approach. like Alice and Yoshida did, and they did sequence the transcriptome of euglenoids in so many different conditions, and they found so many interesting things. And they also highlight that euglenocellular process involve lots and lots of the post-transcriptional level of regulation. By that, does mean using only genome and transcriptome data might not be enough to understand the holistic metabolic capacity of euglena. So what I'm trying to do, I'm trying to understand the metabolic network of euglena a little bit more. And if you think about metabolic network, it will look something like this, telling us where things are and how to get there, comparable to the city map. This is a city of Chiang Mai, where I'm living in, and I think it's look pretty much like TCA cycle. Maybe it's not, but anyway, the map just telling us where things are and how to get there. But whenever you combine the data of the flow of the metabolite into this metabolic network, those data is called metabolic flux. It will tell us a lot more comparable to traffic map. If you look at the traffic map compared to just a normal map that I showed before, the traffic map now can tell you how long does it take for you to get from one destination to another destination, right? And also tell you which route is pretty much free and which is blocked. And if you want to alter it, or if you want to modify or fix the city, which part could be the target. And that's basically what I'm trying to do. I'm trying to create the traffic map of Euglena metabolic network using two different techniques. The first one, I'm trying to predict the metabolic flux using flux balance analysis, which is the technique that based on the stoichiometric of the genome scale metabolic model, starting with the genome and then creating the mathematical model that able to optimize the solution for the objective function of interest, which is usually the biomass production. Another technique that can be used to measure the metabolic flux of euglena is the 13C metabolic flux analysis, that which is the technique that based on the isotopic labeling. The idea of this technique is that you just provide the system or the cell with the label substrate, usually the isotopic label substrate. And when it goes through different pathway, the substrate itself got fraction recombined in a specific way. And you can measure those redistribution or rearrangement of the isotope atom using GCMS or NMR, and then fit those data into the metabolic model, and you got the metabolic flux value. So the objective of this project is to apply both 13C-MFA and FBA to euglena to determine the metabolic phenotype of euglena under the range of respiratory substrate, which include glucose, acetate, ethanol, and pyruvate. And I also took the advantage that we can bleach out the chloroplast of euglenoids and it's still vital to see the impact of absence of its chloroplast in glucose metabolism as well. So the first thing I did, I created a genome scale metabolic model of euglena, even though it's not created but from genome, as we don't have a complete genome yet. It's working in the same sense of genome scale metabolic model. So I think it's still okay calling it genome scale model. So what I did, I used the available transcriptomic data from Alice and Yoshida, and I created a draft and make it metabolically make sense, and also the mathematical makes sense. And then I refined them with the known knowledge of euglena metabolism, including enzymatic study, and also the subcellular localization that I predict and review. And then I got the model that I think could work. And this is how it looked like in here. And my model include about 831 reaction, which include 956 metabolite. And this is a subcellular localization that I predict and review that been integrated into the model as well. And after I get the model, I test whether it do things that I wanted to do, which is the growth of euglena under the heterophic condition. So I provide the model with the different substrate that I mentioned before, glucose, acetate, ethanol, and pyruvate. And turned out the model was able to generate the objective flux, which is the biomass production. So this showing the potential of using the model to explore the metabolic operation of euglena. So let's jump into those. The first part that I'm going to talk about is the metabolic phenotype under glucose condition, or when you grow the euglenoids in glucose. As I mentioned before, I used two different strains, the wild type one and the bleach one. And before starting with the flux analysis, I measure the growth rate, cell density, and rate of glucose uptake, and also biomass composition of the cells. And turn out all of the cell density specific growth rate that calculate from the cell density itself is not significantly different. Also, the rate of glucose uptake as well. And for the biomass composition of euglenoids for both strain, the wild type and bleached one, the proportion between protein, the overall carbohydrate, lipid, and others are not significantly different. But if you look at these two dark blue and light blue here, you will see it's quite different, right? But that's just the insoluble carbohydrate and the soluble carbohydrate, which are arguably quite interchangeable. So I used the biomass composition to constrain my genome scale metabolic model and perform the FBA, flux balance analysis. And this is what the FBA and the model predict metabolic flux to be. I want you to focus on the thicknesses of the arrow here. As you can see, them not much different from both wild type and the bleached cell. And in the wild type cell, the FBA predict no reaction through any of the choroplastic reaction at all. But when we perform the 13C-MFA with the cells, it's showing something quite different. Overall, the bleached cell showing that there's a lot more substrate go through glycolysis and also T-cell cycle. This is central metabolic pathway compared to the wild type cell. And the function of the central metabolic pathway, as we know, is not just only creating the precursor for the biomass, it's also creating the energy cofactors. And by creating the energy cofactor, it's also releasing out the CO2 as well. And this CO2 that been releasing out, we can use it to calculate the thing called carbon conversion efficiency, which is showing how much of the carbon will turn into the biomass. And from the MFA or the metabolic flux analysis that I perform, it's showing that in wild type cell have a lot higher carbon conversion efficiency than those in a plastic or bleached cell. So I validate this using different independent technique based on the radiotope 13C. And it's turned out it's showing the same trend. So I think this could take us maybe the 13C showing something that correct. So what we see here is that bleaching out chloroplast elevate the flux through central metabolic pathway, causing more higher CO2 to be released and also more energy production. But if you remember that the growth rate of the euglenoid is pretty much the same, those surplus energy cofactor that have been produced will be used in the non-growth associated maintenance or the maintenance of the cell, showing that bleaching chloroplast, even though it might not affect the physiology that we can observe, it might decrease the metabolic efficiency of the cells. The next one, let's see how the different carbon composition of the substrate affect the metabolic network. So the substrate I'm going to use here include acetate ethanol and pyruvate. These three substrates entry the metabolic network very similar node. Exactly for acetate ethanol, they entry the metabolic network at acetyl-CoA, which is the same node. But then for pyruvate, it's entry at pyruvate, and then it's go through the TCA cycle. But one big difference from acetate and ethanol is that when you oxidate ethanol into acetate, you got two surplus NADH. And those two surplus NADH is actually quite a lot, considering if you have 100 mole of ethanol coming into the cells, that means that 200 surplus NADH. And that means lots of energy that the cell can utilize. And it would be interesting to see how it would look like when we measure the flux rate. And then I measure the biomass composition, and it's turned out for three substrates, the biomass composition are pretty much the same. But for the growth rate, the ethanol have the highest growth rate compared to acetate and pyruvate. Let's jump into the flux map. There's lots of detail going on here, but I'm just going to highlight something that easy to understand. starting with the pyruvate. The pyruvate going into the pyruvate as expected, and then it's go through the TCA cycle. And then some of them will push back to the gluconeogenesis to produce biomass. And one thing that's quite different from other conditions is that pyruvate, when grow Euglena under pyruvate, this reaction happen, which is the reaction of pyruvate carboxylase. Sorry. And it was not observed in any other condition that I've been observed. For acetate and ethanol, even though they have the same carbon composition, which is 2 carbon, but the metabolic operation when it goes through TCA cycle is quite different. I want you to focus on the reaction here from isocitrate to succinate. In acetate, it's pretty much go through to generate 2 NADH and release out some CO2. But in ethanol, the reaction from isocitrate to succinate is reduced like significantly, and it could be the result from those 2 surplus NADH that generate at the very beginning of the processing of ethanol. And by reducing this, also allow more substrate to go through the siloxylase cycle, which means that more substrate can be pushed to the gluconeogenesis to generate the biomass. And this phenomenon is only observed in Euglena, to my knowledge, even in yeast cell that produce and use the C2 carbon quite a lot. The interchange from the TCA cycle and glycosylation very freely and naturally like this haven't been observed. And that could be the reason from its subcellular localization that glycosylation has been reported to locate in the mitochondria, the same location as TCA cycle. So from those 3 flux maps, it's showing us the that even though we have the same carbon composition as a substrate coming into the metabolic network, the metabolic phenotype can be drastically different depending on the redox potential of the substrate. And Euglena metabolic network can selectively use the specific enzyme to facilitate the metabolism of specific substrate as well. So from all the flux that I show you, one thing that's very highlighted up is that the metabolic network of Euglena has very high flexibility, that it can adjust accordingly to whatever substrate, and not just only because of the rich in enzyme resources. I haven't mentioned about any other isozyme, but from what I've been doing, that's also lots of different use of isozyme in the metabolic network as well. And also the subcellular localization that also contribute to the metabolic flexibility of Euglena. So the future direction that I'm aiming for is that I'm trying to improve the genome scale metabolic model of Euglena. Whenever we have more data on it, I will integrate it into the model to make it more comprehensive and more complete. And we try to use them in autotrophic condition and other condition to maybe target the production of high value product and metabolic engineering in the future. And it would be interesting to see the mitochondrial physiology of Euglena as well as the mitochondria of Euglena. Very intrigued me because they have lots of function going on compared to other organisms. So I would like to thank all of my supervisors, Professor George Radcliffe, Professor Nick Kruger, Dr. Alice Ono, and the Development and Promotion of Science Technology Talent Project and Department of Plant Science at the University of Oxford. Thank you.
Great, thank you. Thank you for a very interesting and informative and metabolically complex presentation.